most important thing is that Sara has agreed to give our colloquium talk today. So I'm really enchanted that you are here and that you will give this talk. And I will take a bit of time to walk us through who Sara actually is. Some of us know very much so. Sara is a Canadian-American astronomer and a planetary scientist from Toronto. And we are now here in Austria, in Graz, and she's part of the Synergy Grant, which includes the UK, now Austria, and the US. So it's a truly international setting, I shall say. Sara got her PhD from Harvard under the supervision of Dmitry Zasilov, and some of you may know who that is. And then later thereafter became senior staff member at the Carnegie Institute in Washington. Since 2007, Sara is at the MIT, first as an associate professor. I know you know this all, but you just have to go through this. But now for physics and for planetary science. So she has 2 professorships. And what I saw when I got the first email at some point from you, you always put them into your name prescription. I think that's a very good thing because you know what you have achieved. So this is a very good example, I think. Sara is now full professor and chair at MIT, and this since 2010. So there's a lot of work and a lot of achievement here now. And she received various prizes which started her career paths. And 2 of them, I just named 2 of them. There's the Order of Canada in 2020. Maybe you want to say a few more words about this because I try to figure out what it is. And then she also received the Kavli Prize in 2024. And most of us have followed this. And of course, it's an utter pleasure to see you receiving that prize. And yeah, just a great example. And lastly, I would like to mention that Sara and her team have proposed a vision called Venus Life Finder, which is due to launch next year in 2025. At least the web tells me, but you're probably more, you can tell us more details. So with these couple of details of your CV, of course, there's much more. So please go and read up because I think Sara is a fantastic role model and has lots to give to all of us younger people. I would like to welcome you on our stage for your presentation, and we are looking forward to it.
Thank you very much. 5 years ago, I was part of a very controversial discovery that shook the world of science, planetary science. How many of you have heard of the phosphine on Venus report? I'll come back to that a bit later. But I just want to say that the search for signs of life beyond Earth, it's a tricky subject. And here's where I hope to give you all the pros and cons and the hard things and where we hope we're going to end up soon. So the real story actually begins billions of years ago, when a simple life form figured out something very clever, how to harness energy from the environment, that is photosynthesis. And these cyanobacteria ended up generating oxygen as a waste product, which over billions of years filled our atmosphere. So we all need oxygen to breathe, but it turns out that oxygen is so reactive, it shouldn't be in our atmosphere at all, unless it's continually produced. So I'm sure many of you are familiar with this idea that oxygen is a biosignature gas. It is definitely one of our favorites. But what you may not have known was it was basically 100 years ago that a renowned astronomer, James Jeans, that's like genes escaped, Jeans theorem, actually in his textbook, this is the first we can find of oxygen as a biosignature. He wrote about oxygen. He also, it was just like 1 page. And he said we should look for oxygen on other planets, solar system planets. And he even put an upper limit on Venus. He put an upper limit to the amount of oxygen he could find from Earth, and said, well, at least there's no complex life because our complex life needs oxygen to survive. So there's so many exoplanets out there that nature has given us something absolutely incredible. If you dig down, you'll see its planets exist in almost every mass, radius, orbit, and temperature that is imaginable. And this is crazy for us to think about. Our solar system might be very rare. Nonetheless, I just want for part of this intro for you to think about all the nearby stars. And I love to think if there's a planet orbiting a nearby star, and they have the kind of telescopes we're hoping to build, and they look back at Earth and they will suspect there's life on our planet. So that's like both the intro and the takeaway. not by big things like the Great Wall of China, not by pollution or city lights, but by oxygen, a gas that is here, it shouldn't be here in these high quantities, it's so reactive, it shouldn't be here at all. I mean, we all know if those aliens are there, they're probably fighting over data analysis, attribution, whether or not it's really from life or not. But that's part of the story that I'm going to tell you about. So I am going to tell you about exoplanet atmospheres. I'll talk to you about the small star opportunity and how we can find signs of life on exoplanets. So some of this will be introductory for you, but I assure you there's going to be new things, and I'm trying to make sure everyone can understand the large majority of this talk. So everyone's heard of a transit, right? And from this, real data is so stunning. It's the relative brightness as a function of time. And this was one of the very first things the James Webb Space Telescope put out for us. This stunning light curve. And you all know that the drop in brightness, it's from the area of the star compared to the area of the planet, right? Pi r squared planet over pi r squared star. And at the top, it's showing you an artist's conception of a very exaggerated star and planet. We don't spatially resolve any stars other than the sun like that. And see, the atmosphere is so big. So the idea for exoplanet atmospheres that the main way we observe them today is when the planet transits. And now look at the star, look at the planet now. With an atmosphere, the planet looks a tiny bit bigger. That's shown by that cartoon. So the goal is to look at the exoplanet at wavelengths where the atmosphere is transparent, and the planet will be a certain size. It will be the size of that dark disk. And then when we can look at a wavelength where the atmosphere is very, very strongly absorbing, we will see the planet looks a little bit bigger, the area is bigger, the drop in brightness is bigger. Everyone with me on this? Okay, because we are going to try it. So what you're seeing here is another exoplanet, WASP-39b, one of our most studied planets, thanks to the James Webb Space Telescope Transiting Exoplanet Community Early Release Science Team. And this is in the infrared. And what you're seeing here is the relative flux normalized to 1 and the time from mid-transit. So it's a transit light curve. Now the planet pi r squared is blocking out about 2 and a bit % of light. And the black is the best fit to all the data over all the wavelengths. The observers here have kindly separated it out for us into colors, or wavelengths rather, and you can look at them. You can look in the top right where it says W, that's wavelength in microns. And your job is to see where, if anywhere, the points are going below the best fit. That means the planet is looking a tiny bit bigger. Does everybody see this? And you can shout out what wavelength.
4.5.
4.5, yes.
4.4.
4.4. So to make sure everyone sees this drop in brightness.
Ready, steady, go.
All right. Well, wow, let's let this land. By eye, you have all seen evidence for an exoplanet atmosphere. That is a triumph for all of us engineers and scientists that we can see this. And it's so amazing for exoplanets that we have this level of data. So what we do with this data is we'll take every transit and measure a depth, put an error bar on it. So every one of these points is a different wavelength. So each one of those colors or wavelengths translates to 1 point on this graph. And you can see its amount of light blocked as a function of wavelength now. The white is data, the blue is it's a best fit model. Now what you can see here is it's very different from a straight line. Honestly, the old data when I used to give talks, you'd be like, I'd say, yeah, it's different from a straight line. And people be like, not really sure. In this case, it's unequivocal. The planet looks a tiny bit bigger. At wavelengths, we said 4.5, we had 4.4. It is definitely bigger. And this wavelength is where carbon dioxide is strongly absorbing. So the people in the audience, this is boring to them if you work on this, because we're seeing a lot of giant planets have strong signal there. But interestingly, carbon dioxide is not the dominant form of carbon. It is just carbon dioxide is a very strong absorber, right? And for the non-exoplanet scientists or non-atmospheric scientists, think of it like a skunk smell. A tiny bit makes a big difference. So this is a hot giant planet. It's not going to have life on it. It's really the best, one of the best we have to offer in exoplanets. Here's another graph of the same planet. Now it's a longer wavelength. So all the ups and downs, the planet's looking a tiny bit bigger. They're all different gases. And this is really the introduction to you, that we can watch the planet change size slightly as a function of wavelength, and we can map those onto gases in the atmosphere and learn about the exoplanet atmosphere. That's how we hope to find signs of life by way of gases in small planet atmospheres, cooler planets, that we can find biosignature gases. And that's what most of my talk is going to be about. But I thought I would pause for a moment here because, believe this or not, one of the reasons I have all those awards is when I started working on exoplanets, pretty much no one else was. I worked on exoplanet atmospheres last century, finished my PhD in 1999. And it was really a big question mark for people, even those on my own thesis committee, as to why I was working on this. And people were just saying, well, we'll never detect their atmospheres. There were about 20 or 30 planets known, and I went to start my postdoc, and I kept getting asked, what's the next big thing? Which is translation for, if you're really bad at small talk, that's a good thing to ask people. What's the next big thing in your field? And I said, transit, because we had no transiting planets yet, but there was going to be one any day, right? Because the probability to transit is a certain value, and we had a certain number of planets, so it was going to happen. That's when I started working on this idea of transmission spectroscopy. So I'm just trying to tell you this because even though during my postdoc, observers took my idea, measured the first exoplanet atmosphere, it literally was not enough to get a job because people called it a one object, one method success. They thought the field would go nowhere. Rightly so, because the stars are so big and massive and bright compared to the planet. People didn't have the nerve or the vision to really make it work. Now, I was a postdoc. I wasn't like I was so strategic and visionary. It was just kind of, it felt like the right thing to do. I just kind of went from project to project. And this was one of them. But the reason I'm telling you all this now is to set up the theme of my talk, that the line between what is considered mainstream research and what is considered crazy, that line is constantly shifting. So all this work now is so mainstream. It's probably too mainstream, really, but it's wow. And I'm not sure if you're ready for my talk, but a lot of it is really on that side of crazy. We're going to come to that a bit later. So for now, I did want to give you some, okay, it's crazy. I just forgot. I feel like it was partly an assigned task to go into a bit of detail. So I took it upon myself to just share with you where we're at in the field now, and especially if you want to work on atmospheres, I just have some tips related to our challenges. So one thing about this light curve, I know it's not necessarily the best fit. Don't worry about the blue line. We'll get to that. But in all the models we means hundreds of people have been making, we didn't get it all right, actually. And there's a little bump that showed up that people were really surprised about. This little bump here. Can you see that? The white points? Sulfur dioxide. It may not mean a lot to you, but typically in the giant planets, they're hydrogen dominated, right? They're mostly hydrogen and helium. So kind of to first order, we expect all the elements to be in their most hydrogenated form. So you expect sulfur to go with H, that's H2S. But instead we found a bunch of SO2. So that was one thing, like, okay, we're gonna have to figure this out. So I just wanted to show this to you. It's kind of supposed to be for fun, and I highlighted a simplified schematic. And what this person is doing, Syed al 2023, was he had to introduce photochemistry for sulfur. As you literally write down every single form of sulfur. So you have, I can see some astrochemists nodding because they understand what it means. But essentially, look at all the different molecules you can get. And all the arrows are the different chemical pathways. And you have to work this all into a computer code that couples to the rest of your atmosphere model. So what I see today as the frontier of exoplanet atmospheres is literally photochemistry. It's like a new branch of astrochemistry in many ways. Some of the reaction rates we don't really know very well. So it's definitely about chemistry. And I did have another complicated chemistry example for you. And that was actually, it goes back to our planet Venus, which we'll come back to. So in Venus, I think most of you know it has a massive carbon dioxide greenhouse atmosphere. But if you were to work out the photochemistry, like people did in the 80s, they found it actually shouldn't. Because carbon dioxide gets photodissociated and it gets separated. And it should recombine to three things. CO, carbon monoxide, CO2, carbon dioxide, and O2. But it's all in CO2, so what's happening? Well, the people who made these models said there has to be an unseen catalyst, something that forces it to go back to CO2 and not to equal parts these other things. I'm just trying to get you to think about exoplanets now. We're talking about Venus. And later people found these molecules are like chlorine molecules, HCl. But I'm so like, not enough maybe, but it's just so astonishing to me that our own sister planet Venus has so many mysteries, detail and photochemistry that we might find really hard to deal with. I had another more complicated example. I just wanted to show it to you because I'm trying to impress upon you the complexity of atmospheres and the kind of limited data for exoplanets that we have. This is showing you height above the surface in kilometers on Venus. This is an unfortunate term for the volume fraction. So you're supposed to look at sulfur dioxide again in this planet. This is Venus's atmosphere. This is a model. And it shows you it's about 1 part in 10,000. It's like a volume mixing ratio. So on Venus, there are volcanoes, they give off sulfur dioxide, it rises. And this makes sense, right? That your model would say somewhere very high in the atmosphere, the UV light from our sun breaks up sulfur dioxide. So that's kind of like what the code shows. But the data shown here, which is from both ground-based measurements and sounders that went to Venus in the past decades, it doesn't match at all, right? I mean, look at this, it's so crazy. But what people do is they just, it's still not really known why it's so different. That's the bottom line, that there's this major depletion of sulfur dioxide in the atmosphere. And it's kind of another story. We worked with Paul Rimmer on why some people just fudge their models and tell us what their fudging factor is. Others of us are like, maybe there's something in the cloud particles that's changing the chemistry. But I just wanted to impress on you that planets are complicated, very complicated. So if you were to take a class on exoplanet atmospheres, you'd have to learn the basic equations, and we all kind of wish our field was simple. Like we could write down 5 equations, have 5 unknowns. That's what I've written for you there. And it would be lovely if we could just take a matrix and invert it to get the answer. We have our equations of conservation laws of radiative transfer. We have chemistry, and we want to know the temperature, pressure, and radiation and chemical composition throughout the atmosphere. Like in principle, it seems doable mathematically, but the problem is I gave you the one example of photochemistry. We have another example I should mention here, clouds, microphysics of clouds. These are insanely complex, and we really struggle to know how to capture this whole problem. And it's definitely a challenge for us because we have not only all the known microphysics, I think I gave you a glimpse that we definitely are going to have unknown photochemistry. But from this mathematical viewpoint, we basically don't have well-defined boundary conditions. How our layers are connected to us is unknown. And this is a big problem. If you work in another field other than exoplanets, this would be like a nightmare to you. You would not want to go here. But what we decided to do quite a while ago was just not worry about this and just say, what we're going to do? We are going to capture every single possibility that the data tells us. We're going to try to capture every possibility of the underlying physical parameters that the data tells us. How many of you have worked on something like this? Like poor data, sparse data, lots and lots of options in the model. Yeah, essentially we have so many free parameters in the model. And this is kind of this is the heart of why it's going to be so hard to find signs of life. Okay, so stay with me here. I had much more I could say on this, but instead I have this gorgeous cartoon I got from Ryan McDonald. Just in case you haven't done this, you can call it like an inverse method. I'm only going to give the simplest version of it here. So we can't invert the matrix because our equations are nonlinear, but we can still do an inverse method. And in this case, we're showing you the amount of light blocked as a function of wavelengths. Remember that from the real data? And these are fake points. This is a fake model. We're assuming this model only has temperature structure. We're trying to understand the underlying temperature and abundance of water. One second, this decided to stop working. Oh, because it wants me to. Okay. Good model. How do you like that model? Bad model. So our first paper on this, we literally called it the million model approach. We just generated millions and millions of models, ask which ones fit the data, which ones don't fit the data, and come up with a underlying physical parameters that fit the data. It's so sophisticated now. You can use many different ways like Markov chain Monte Carlo, nested sampling, there's machine learning. There's a lot of ways to handle this problem. I think a lot of you have done this or seen this somehow. But in exoplanets, it's kind of the main game in town right now. And in this example, Ryan McDonald picked for us, temperature 600 plus 120 minus 175. It's not far from the truth. We have ginormous uncertainties in the underlying physical parameters. H2O4 plus 2 minus 2%. I mean, that is kind of where we're at. So although we have these gorgeous spectra, we can identify molecules. It's really hard for us to say something specific about them, actually. So I have a little cartoon for you made by myself and Louis Wellbanks and one of his students. And it's just a cartoon to capture something so complex in the simplest way possible. We have our transit, we talked about that. We have our observation. Now let's imagine we've retrieved, we have two molecules, molecule X, she's calling it, and biosignature Y. So those are just our transmission spectroscopy. This cartoon looks great, right? We've got data points. And we do this retrieval, an inverse method, and we get a probability of the abundance of certain gases. And then yay, find signs of life. That's kind of what we hope would happen based on all the complex machinery. But what ends up happening will be no surprise to you. It ends up being way more complex. Look, this star has activity. And just getting from our observations to our transit light curve, it's very messy. The reveal team here, we're working on limb darkening. So even going from your observations to your spectrum, there's biases in how you subjectively treat parameters or how many free parameters you add to get those from the data. So in the end, it's actually not very clear at all. And what we're seeing in our field is people doing these inverse methods will get different answers from the exact same data. Essentially, what it's boiling down to is, just to oversimplify the problem, we have too many free parameters for our models. And what ends up happening is that our model retrievals are so sensitive to structure and the noise. And even with the same data and as similar model as we can have, your model could give a different answer from mine. And now we're so confused that we're getting different interpretations for the same planet. And this is like a separate topic. You can have a whole other talk on the mini Neptune planets. What are they made of? We're getting completely different ideas. Some of people think they're water worlds made of 50% more of water. Some people will say they're giant rocks with a massive hydrogen helium envelope acting as a greenhouse, making it not just a massive rock, but a magma ocean rock at that. So I just wanted to let you know where we are in the field here. I gave you two examples, very complicated photochemistry, even hidden things we might not have evidence for, using Venus as an analog. And this whole free parameter, too much complicated physics and chemistry for us to really know how to handle. But I can assure you, a lot of people are working on those. And that's kind of where we're at with exoplanet atmospheres. All right, so mostly those were about giant planets. And it turns out for atmospheres, we're not able to study an Earth around a sun using transmission spectroscopy. If it were an undergrad, I understand classes are almost in the city, but if it were an undergrad class, we could work out why. Because the ratio of the area surrounding a small planet compared to the area of the star is just too tiny. It's so tiny that we just cannot measure those atmospheres. And that's, I gave you some numbers there. I put some math if anyone wanted to browse it. So the whole field now is funneling towards these small planets and small stars. And I put the numbers for you at the bottom. We can study, yeah. And how many here, I know that half of the audience knows TRAPPIST-1. Has like everybody heard of TRAPPIST-1? It's literally the favorite star right now. It's a tiny, tiny star. Any smaller and colder wouldn't be a star. And it's like getting outsized attention in the field right now. So I wanted to just share one thing that the Reveal team is working on and try to explain it for you, because this is our main challenge that we're facing right now in trying to study atmospheres of small planets transiting small stars. And what I'm showing you here is transit depth as a function of wavelength with these up and downs. But remember before, we all agreed that the drop in brightness is the area of the planet compared to the area of the star. So for this argument, we're pretending that the planet has no atmosphere, which we think is true for some planets. So if the planet has no atmosphere, pi r squared is constant, but now we're adopting a star that has a spot. Now pi r star squared is changing as a function of wavelength because the spot itself is changing as a function of wavelength. I just want you to think about that for a moment, because this is like the main thing in the field right now. And that's one of the reasons why the Reveal Consortium exists. So this change in transit depth is not due to a planet, which we're assuming to have no atmosphere. It's due to a spot, which has wavelength dependence to it, because the apparent size of the star is changing as a function of wavelength. And so we have our simulated atmosphere, our simulated star activity, those are all mixed together. And that's the biggest problem of our day, is to unpack it now. And one of the reasons we do think, well, what we propose to do for our synergy grant is to disentangle these using Sasha's MHD first principles models of stellar activity manifestation on the surface. So that's why I've been visiting. Here's some of our team. And you'll have plenty of chances to interact with everyone. If you're here, and for Reveal also, we have a couple of other people, including Ignacy Ribas, who has his own ERC advanced grant. So if you're part of this consortium in here, can you just raise your hand so everyone can see what a large group we have? This isn't even everybody, but these are all for this. So good work, team. One thing from the meeting I wanted to share with you, because it just blew my mind. I honestly couldn't even handle this. That we have some cartoons of what we think TRAPPIST-1, I'm saying we, but this is really Ben Rackham and the first author, Ratke, are putting out a paper. It's supposed to come out this Friday. And they've done something very clever. What they've done, I have to back up for a second, so excuse me for going back in time. They have taken observations of TRAPPIST-1 using TRAPPIST-1b and c, which are both believed to not have an atmosphere. So they're literally doing this. And they're finding that it appears that like 40% of TRAPPIST-1 is covered by something dark. That's a lot, 40%. So it could be this, we're calling it the salt and pepper star. Like, can you imagine if it was like that, if this is granulation or flux tubes or miniature spots? But it's also consistent with this picture with these ginormously large, kind of continuously covered polar spots. So this part of the talk was to impress upon you contamination and how we're learning so much about stars, and each one of those like 25 people will have a different phenomenal thing they took away from our meeting, because it's rapidly progressing to be using planets as probes for the star itself. Okay, so we're now going to switch gears to biosignature gases. But for now, you have to pretend we're like 5 or 10 years in the future, when we've wrestled our inverse methods to get more solid, agreeable answers of what's in the planet atmosphere. We have completed our spotless and our synergy grants, and we have managed to get rid of a lot of the stellar contamination. And then we can ask ourselves, what gases should we look for? It's true that oxygen is the dominant biosignature gas on our planet, but we've had oxygen only for, I can only say this to a group of astronomers, only for 1 billion years. Because it took a long time for even though life has made oxygen for billions of years, it took a long time to saturate the sinks and to kind of accumulate. And so we can ask ourselves, what other gases? It turns out there's thousands of gases, not just oxygen. But what I did was I uncovered a little factoid that every gas in Earth's atmosphere that is present to the part per trillion level, with the exception of the noble gases. So every gas present to the part per trillion level is made by life, even though they all have a dominant source that is not life. So maybe with the exception of the astrochemists, but I could challenge you to pick a gas you know, and we can see if it was made by life, and I'm sure it is. Because out of the 14,000 of gases or so that are, out of the 14,000 molecules that are room temperature are in gas form, literally 1/4 of them are made by life. The mold in your fridge, the smell coming off of the pine forests, most are in tiny quantities, but there's just so many gases. And I went on a giant kind of, I don't know what you want to call it, like, it's like when the squirrels in the fall, they're collecting the nuts to store for later. Like, you don't know why they're overdoing it. We went on a giant tangent in 15 years to collect all the information about every molecule that's in gas form at like habitable conditions. We found out which ones are made by life. We curated these databases. We're working on it now. We actually have a lot of threads to pursue. But for our purposes here, we're asking ourselves which gases could be useful biosignature gases. And I had some more technical slides I cut out for the interest of time. I just tried to boil it down to the main thing that matters, and it's photochemistry again. So my message, it's not one I like, but it's the real message that to study exoplanets, we need to understand chemistry, photochemistry specifically, because it turns out that all molecules, right, all trace gases, life or not, they float into the atmosphere and they get destroyed by photochemistry. Either radiation from the star itself breaks them apart, or there's like a cascade. Usually the radiation makes OH. On Earth, we call OH, the radical, the garbage eater of the atmosphere. It destroys everything. And so what we're finding is that any gas that you want to put forward as a biosignature, you've got to work out the photochemistry. You have to ask yourself, how much needs to be produced to withstand photochemistry or other sinks. And so I put a little chart for some of them, surface flux on a log scale. And for some gases, like here's what oxygen is on Earth. So that, just look at the relative scale. Our life is putting out this much oxygen, this much methane. And for a few of the gases we've studied, we just need way more. So we have to ask ourselves, you know, that's really our main problem is photochemistry. Okay, so I just have the summary. It's nothing neither here nor there. You can add your favorite gas, but I've written a review article to come out next year. And we have this table showing you gas. And we have our 3 key questions. Does it have a unique spectral feature compared to background gases? You know, does it have a reasonable production rate, given the sinks like photochemistry? And does it have false positives? So this is a bit of a contrived diagram. You know, you don't have to take it too seriously. But do you see any good biosignatures here? I've only given you a minute. You didn't have time to think about this on your own. I'm just giving you a summary from a bunch of papers listed at the bottom. And so far, really, in terms of a gas that can be produced in large quantities, that can overcome its sinks, that has no false positive, that has unique spectral features, we're definitely coming up short here. And so this is really something, it's a struggle. You know, I didn't want to come here and be sorry it's over. But there's a lot of work to be done, but it's not clear to me that there's going to be some obscure gas produced in way more amounts than oxygen is on Earth. We really need to get lucky here. So out of all these gases, which one do you think is my favorite?
KH3.
See, I put brackets, yeah. I actually need to get rid of that bias, but yes, phosphine is my favorite biosignature gas without question. It is a very unique gas. We have almost no phosphine. Phosphorus wants to go with oxygen. So we have phosphates, they're not gas. Also, phosphine is highly thermodynamically disfavored. You won't just get it like lying around or like out of a volcano. It's very rare. It's only associated with life on Earth. There's very few gases we can say that are only associated with life and not some geochemistry or photochemistry or something else. And phosphine is one of the only ones. We make it as pesticides. It's found in oxygen-free environments. So phosphine is the one to remember. Okay, so far in this talk, I'm sure I've convinced you we have an established method to observe atmospheres. But at the bottom, we have stellar activity contamination, this tension between the number of free parameters and the number of data points. We have known rocky planets in the habitable zone. Pretty much everyone heard of TRAPPIST-1. And we have a list of potential biosignatures. So it sounds like we're in pretty good shape, right? I mean, we can work through most of these problems. That's what I thought. I was working on this for my life goal, and I see a growing number of younger people, it's their life goal to find signs of life on another planet. And then what happened? So now I'm going to unpack very briefly the story of phosphine on Venus. And while I'm doing that, I need you to think about an exoplanet. Okay, so I was working on phosphine as my favorite biosignature gas, and it turned out Professor Jane Greaves was also working on phosphine. And you know the line between mainstream and crazy? It was definitely crazy to search for signs of life on Venus, but that's what she was doing. And to her credit, she'd also found that phosphine is a wonderful biosignature gas based on our own Earth. She proposed to observe it, was obviously rejected many times, finally got time. And somewhere into her study, someone connected our 2 teams. So we went to work with her to help her with proposals and interpret data. So we made this announcement in the fall of 2020. And we never said we found life. We just said we have reported detection of phosphine on Venus at the part per billion level. We said we worked through 100 pages of chemistry, but of course it's quality, not quantity. And we've showed that all the things we know about on Venus, there's no way that it can be, we said that out of everything we know, it's not made by any volcanoes or lightning or meteorites or anything we can work through. So it leaves room for life on Venus. That's exactly how we put it. And what do you think the reaction was? There was immediate anger and backlash for lots of reasons. So remember, we're thinking about an exoplanet now while I tell the Venus story. First question, is the signal real? Great question. Obviously, not the obvious first question if you're a scientist. Well, people took the data and looked through the data. And this data comes from radio observatories, James Clerk Maxwell Telescope. It came from ALMA. And Venus is bright, too bright, actually. It's spatially resolved. There's a lot of noise. People didn't like how we handled removing systematics. People looked at the data again quickly, though, and they actually did not recover the signal. Some groups actually did recover the signal, and there's a lot more there. But let's just for a moment assume the signal is real. I mean, there's still controversy over that. The large majority don't think the signal's real. There are people who do still find the signal, independent teams using the JCMT data. Let's assume the signal is real. Remember, we're thinking about an exoplanet. Is the molecule correctly identified? Out in the radio, where these observations are taken, this is the ground to first excited rotational state of phosphine. Usually things are pretty separated and you don't have overlaps. Obviously you'd want more than one line, but there just weren't any other lines. Unfortunately, there is a close overlap, sulfur dioxide, which we've talked about a lot. It is a planetary gas. But we did have some near simultaneous data, which showed that it shouldn't be sulfur dioxide because it would have had to increase in value too much to be explainable. Nonetheless, it is still a possibility. Let's imagine for Venus or an exoplanet, we could accomplish the first two. Is it the signal's real? It's now correctly identified. So now what? We have to agree that it's made by life and not made by an abiotic process. But people still won't agree on that on Venus. They come up with ideas. I'm trying to be neutral. It's going to be hard, but like a volcano can make something, but Venus would have to have way more water in the mantle. Remember, we need hydrogen to make phosphine. And it would have to be so explosive, like more explosive than anything on Earth to shoot that phosphine up into high in the atmosphere. But there's still a ton of back and forth on phosphine. Oddly enough, the Pioneer Venus, NASA's mission from the late 1970s, has evidence of phosphine. So this story is going to keep going on. And if you were interested in unpacking it, we have like a living table showing you we have one for observations, one for interpretation. Literally every paper we respond to. And I'm not going to walk you through it just to know it's there. And it's really Professor Jane Greaves and team literally put out a paper. It's a full-time job, like responding to every criticism. And so this controversy goes on. So we have these, I think I convinced you we were in a good place, but this whole phosphine thing brings a crisis to the search for signs of life elsewhere. Because if we can't agree on Venus, exoplanets are still going to have a weak signal, perhaps the wrong attribution. We don't know a thing about exoplanets in terms of like how to know about the geo and photochemical processes. So can we robustly find signs of life? I mean, that's a question for everybody here. We're all astronomers. We're all kind of on the hook for making the search real. Okay, so while we're going to mull this question over, you know how sometimes like when you sleep on it and then you wake up like you know the answer? I want you to mull it over because I have to take like a mini digression to what happened to me. This is my story, but it's a lot of people's story now. And this is where I wanted to invoke the line between mainstream and crazy is constantly shifting. Because I have something so crazy to tell you, I can barely believe it myself. And it starts with Venus. You all know it's too hot for life of any kind at the surface. But just like on Earth, if you hike up a mountain, it gets colder and colder. So too on Venus. So 50 kilometers above the surface, it is just the right temperature for life. And that's why half a century ago, Carl Sagan first put the idea out there that there could be life on Venus. But this life would have to be 50 kilometers above in the clouds. It would have to persist aloft. And what's interesting about Venus is that the clouds are permanent. They're covering the planet always. They're vertically thick. They're always there. We have life in our clouds on Earth. And this life gets swept up off the surface, goes into the clouds, stays there for about a week. So it kind of sounds possible. We think Carl Sagan didn't know that the Venus clouds are not made of water. They're actually made of concentrated sulfuric acid. This is such a nasty substance. I don't know if I have time I just have this quick mini story to like illustrate how crazy the substance is. Now we're going to dial back to the pandemic summer, where I was working on the Venus work and we were thinking about a mission to Venus. And at the time I had a 17-year-old son at home. Some of you might have been 17 during the pandemic. Remember, there were like no jobs, nothing to do. They closed the sports fields. You weren't allowed to like even basketball. They took the nets away. So I put the 17-year-old on the Venus project, and it was like an online project. Well, he finished that project in like a week because he discovered like a single source database that had all the answers. So he pushed to do real experiments. So I said, okay, let's try it. So we found out, first of all, you cannot order large amounts of sulfuric acid to your house. Because remember, labs are closed, businesses are closed, but you can actually order a lot of little bottles. I'm sure if like someone's watching this, they're gonna make that not possible anymore. So we ordered a lot of little bottles, set up an outdoor lab to the best we could, because he was still in high school, he wasn't, yeah. And what's the first thing you're gonna put in sulfuric acid? Hot pandemic summer, outdoor laboratory, leafy suburb. I'm gonna have to look at the young, well, not like, I don't wanna say like, yeah. What would you put in? Let's take something, throw it out there. There's no right or wrong answer. I'm just curious. You're trying to understand concentrated sulfuric acid.
It's luck.
Oh, right.
Musk.
Pardon me?
Musk.
Any other answers? We have some nice people, some not as nice people. He put an ant in sulfuric acid. Before I could say, please don't do that. This ant had a seizure and turned to goo before you could blink an eye. So that's concentrated sulfuric acid. Our life cannot survive. It turns out our DNA can't survive, actually. So our life is not surviving. So that's why it's like on the crazy side of crazy, searching for a life on sulfuric acid. But what I'm here to tell you is something absolutely astonishing. We have carried out experiments now that have shown that actually life could survive in sulfuric acid. Not our life, but other life. And just to wrap up the story, said son, he's now 21. He's actually a biochemist studying biochemistry, and he's worked on it. And well, I don't know, have you, okay, there's the astrochemists probably understand organic chemistry, but trust me, no other astronomer or physicist understands organic chemistry. So having someone on your team or in your family who understands organic chemistry, it's amazing. They have like a natural ability for it. So one of his, we put him on a project to put our 20 amino acids, our biogenic amino acids in sulfuric acid. And guess what? We found they're all stable. Except for one. Half are chemically modified, they're stable. We put not our DNA, but our nucleic acid bases, the latter, ACGT, in sulfuric acid, all stable. Protonated, but stable for a year or more. We have put lots of things. We're trying to make a DNA-like molecule. We actually got some money from the Sloan Foundation of the United States of America, and we are trying to generate a DNA-like molecule by swapping out parts. We're not trying to say there is definitely life on Venus. We're trying to establish the science case for going to Venus to look for signs of life. We have a lot of things going on. I don't have time to tell you. I like an entire talk about Venus. It's the talk I really wanted to give, but I have a summary for one other thing. We had some colleagues take lipids with a polar head to put in sulfuric acid. They put these in water to try to make life from scratch. In the series of trying to make life from scratch, you need a cell membrane. And so they put these things in water, the polar head points outward to water and makes a little spherical vesicle. We put this in concentrated sulfuric acid, and the molecules my colleague Janusz Petkowski chose worked on the first try, made tiny spherical vesicle. So I ask you, is he so brilliant that the first thing he picked survived in sulfuric acid and formed a little vesicle, like a cell-sized membrane? Or can many compounds work? Well, it turned out many compounds can work, and not only that, someone had already done this work decades ago, not associated with Venus at all. And they want me to show this to you, which is showing you, this is like a microscope image of the spherical vesicles, showing you scale of 10 micron, 10 micron. It's amazing they're dye, but the dye itself survived in sulfuric acid. Much more complex structures ended up forming that when you do this in water, it ends up being something you have to really encourage to happen. Anyway, there's a lot more I've learned about biochemistry. I'm happy to share it with you during the discussion. I'm so pleased to have my own chemistry lab now, and we have a ton of work going on here. But all this work is just to go back to Venus. And we have these series of missions. We had to rebrand from Venus Life Finder, it was too crazy, to Morningstar missions to Venus. So sometimes Venus shines brightly in the morning sky, and we like to think of it like a new dawn is rising for Venus. What might be of interest to you, we're also trying to change the way we do space science. We as humanity have not been back to Venus since the late 1970s, early 1980s, when NASA and the former Soviet Union sent dozens of probes. We want to have more frequent, small focus missions to Venus. We have our first mission to Venus, the Rocket Lab mission to Venus, largely privately funded. We're delayed till about 2026. I don't have time to tell you about more of our stuff, but our next mission is going to be a small balloon mission. I can tell you more about that in the Q&A. Okay, I'm going to work on wrapping up now, but just wondering if you mulled that question over while we were doing a little diversion. The question is, can we ever claim a robust discovery of signs of life? This is a really hard question, and if you're interested in working on the field, then this is the question that we all need to contribute to. So I'm going to wrap up with a few slides on ideas of how we're going to go forward. The first idea is to make incredibly detailed models of every possible thing we can. I definitely think of Christiane Helling and her career's work on microphysics of clouds, but we would have to do that literally for every other area we could imagine, not just for clouds, but everything, and try to understand everything in detail. I actually think we have to do this, and I'm considering embarking on it, but how else can we weigh in on whether or not there's signs of life or something else? At the same time, I really do think we're going to find biosignature candidates. We'll find a candidate, we won't know for sure if it's right, but we have to do all of this even to know whether there's some chance of it being a sign of life. Another idea is to find an Earth twin. So not the Earth cousins, not the small planets transiting the TRAPPIST stars. And this is another part of our REVEAL project where Andrew Collet-Cameron is working to find Earth mass planets orbiting sun-like stars. And our goal is to go to space. And I put the NASA Habitable Worlds Observatory, Life coming out of Switzerland, and Starshade. Lots of ideas, a few key ideas of how we're going to look at an Earth twin. And it may be easier for us to make it a biosignature planet candidate, knowing more about the planet. Now, it would help to know the targets. It'll save us a lot of money and time on our mission, and we need to get those targets. What we're really going to need to do, and now I'm speaking to the younger people in the room, we're going to need to invest in the ideas that are way out there. Think about 100 years ago, and if someone came up to you and said, we're going to build a space telescope, and this was 1925, no, I mean, that would just be not conceivable. Now we've got to think, what is that thing today? And here are two ideas. I just came back from Luxembourg, where we had the first European Interstellar Conference, a conference that has been going on in the US for a while. If this doesn't make you have the giggle factor, I don't know what will. But I'm so inspired by this conference because for the first time, we heard a lot of details on two ideas of how we're going to be able to better know their signs of life. The Solar Gravitational Lens Telescope will be a mission that goes so close to the sun, it slingshots around, and just as it slingshots, we'll deploy a solar sail and get accelerated. And this solar sail will have to go to 500 times the Earth's sun distance, using our sun as a gravitational lens to magnify a known background object and get a spatial resolution to 10 or 20 square kilometers, magnifying it both in brightness, be able to do wonderful spectral resolution. So we need new ideas. We've got to invest so that this generation, we will come up with the planets with biosignature candidates, but we'll need to wait till later generations to really seal the deal. So to summarize, there are thousands of exoplanets known to orbit nearby stars. We have a mature method to study exoplanets, atmospheres. I tried to emphasize for you the inference problem, or the inverse method of understanding how much gas, what are the underlying physical parameters. We talked a bit about the stellar contamination. Our first chance to find signs of life is with the James Webb Space Telescope, and people will be claiming signs of life. And then you're going to go back and think of the Venus example. Meanwhile, Venus has taken a complete life of its own now, and stay tuned for more info on Venus. So let me just thank you for your attention and encourage you to think big.
Thank you so much, Sarah. I tried, before I hand over to the audience, I think I made 3 notes. So you have touched upon basically everything that what makes at least science for me. That's the ethics in science. Talking about Jane Reeves, I think we have 3-4 people in the room who have been in St. Andrews. 3 of us have been there when Jane made this discovery. Ludmila joined later. So we have seen the storm that appeared, and it was not nice. So I think there's a lot to be learned, also how to trust the astronomical societies. Then you repeatedly have pointed us to how difficult or how necessary it is to walk the path between real new science, so making the next step, or more of the same. And I think this is something all of us have to breathe in, basically. And then my favorite is actually that you came up with the retrieval, because I'm clearly the one who stands on the forward modeling side, where we really try to beat the grid, have enough grid to really carry out a forward modeling. So there's lots to think about from your talk, and with this, having taken the privilege of the moderator of the session, I now hand over to questions in the audience.
I'm curious about mission to Venus.
Second. Possible? Okay, I'm told he doesn't. We don't need it, so go ahead.
I'm curious regarding mission to Venus. Maybe you could tell us more.
First mission to Venus, we call it the Rocket Lab mission to Venus. We have partnered with Rocket Lab, who are supplying the rocket, the cruise vehicle, the probe, and inside the probe is a pressure vessel. This is a very cheap, fast vehicle. There's no parachute, so it will go into Venus and slow down by its own drag, having 5 minutes in the clouds. We were allocated 1 kilogram, and we have one instrument. We call it autofluorescence nephelometer. It will shine an ultraviolet laser through a window and measure the backscattered polarized radiation. We're trying to see confirm if the particles are pure sulfuric acid or if they're a bit different. And also, we're searching for fluorescence. It turns out, so pure sulfuric acid will not show fluorescence, and organic molecules do actually. And so it's just kind of a first mission to show we can get to Venus cheaply, to find some interesting science and move on. And that's the first mission to Venus. The one we're spending a lot of time on now is this balloon mission. We have a whole series of instruments, and we're ramping up this summer to find signs of life on Earth using our instruments.
Yeah, Sarah, thanks for the inspiring talk. Out of your experience with the Venus phosphine story, how do you think we should proceed when find some evidence of a biosignature, let's say? Is this from lessons learned, let's say, at the community level? How should we handle that? Because it's quite likely to come to a storm like the one we had.
Yeah. I honestly don't know if there is a better way. Like, I'm not saying this way was like the best way, but we had okay, let me back up with a bit of background info. So I have been, I'm no longer working on it, but I worked on the test team, and these big missions, perhaps Chaos and Plato do the same, but we put out a different level of data products. We'll give you almost raw data, not quite raw data. We'll give you more and more sophisticated data products, right down to the massage light curves. So on ALMA, they do something similar, but we could look at the data from a much basic perspective, like more raw data, but we had people on the ALMA team working with us. I wasn't part of the data analysis side, so we did everything right in terms of the best way to work with the data. It turned out ALMA had a little mistake, which made our signal weaker than we originally presented. You know, but we never said we found signs of life, we just said we have this, yeah. So it's definitely a tricky one. I think we have to do a better job in maybe not even saying at least room for life. Maybe we've got to stop a bit short there.
Maybe I can offer one more thought on this. I mean, we have seen how it was publicized in the UK. Yeah. And I think this was the mistake. This was the biggest mistake, or this was the mistake in the entire process. Mm-hmm. On the science side, it was, what else could you have done? What the way it was used, and back at the Center for Exoplanet Science, we actually wanted to start a project called What If? Mm-hmm. What do we have to do if we discover life? Roping in philosophers, social scientists, historians, astrophysicists, geosciences. So we had it all there. Yeah. We just didn't get there. Yeah. So
I think it is too inevitable though. Like, we could talk about this a lot, where even if we're very careful with the press, it'll still take on a life of its own, because people love this. Like, the public is behind us. They want us to find it. And so we'll definitely snowball no matter how careful you are. That's my strong belief on that. But I do want to say one other thing. Like, I'll give this talk over and over again. And my message, what I'm trying to do is say, it's not about phosphine. Let's let the phosphine will rage on its own. Like, you know, there's methane on Mars. There's like 3 completely independent, say 4 sigma detections of methane from ground-based telescopes, from a rover, from an orbiter. And do you think the Mars community 100% believes there's methane? So this is going to continue. I'm trying to say, look, the search for the missions to Venus, the life possibility for biomolecules in sulfuric acid, phosphine is just the gateway to those. It's not about phosphine. But still, the conversation keeps going back to phosphine.
Yeah. We had Gabrielle.
Do I speak loud or?
Yeah, speak
Okay.
loud.
On the phosphine question, how many times the detection was repeated? And in the broader scope, how many signs of biosignature should you expect? You said multiple lines would be better. If you observe an exoplanet, how many lines or distinct signs you would expect to have more confidence? Like, because, yeah.
Sure. So, well, one of the things that we liked about our work was we had 2 different telescopes found signs of phosphine. So it was JCMT in Hawaii, like a single dish radio telescope, and ALMA, like the array in Chile. So having with 2 independent telescopes, we thought was really great in terms of different observations. Jane Greaves has since gone back and made more observations and seen it again. So there's that. People tried to look for it at near infrared, but the clouds are really, we think that it's coming from in the cloud layers, and the clouds are prohibitive for that because you only see reflection near infrared. So there's that. So the radio, I'm not a radio astronomer myself, but it seems kind of clean in terms of the way, you know, rotational lines are separated. In near infrared, we have rotation, vibration, and it's a mess. So it could be much harder to assign a molecule, assign a line to a molecule. We could have more than one, but we're still working through that right now, you know? People are even making claims already based on just a single line. It's just messy.
My question goes to the Venus balloon mission. How deep is it flying? And the question I'm asking is, for Venus, it is very difficult to make a 3D climate model, and I'm a 3D climate model, and we don't even know how many circulation cells there are and if they're like 2 on top of each other. And that is very important because you need to also understand the coupling between the surface chemistry and the upper atmosphere. Because we also not only biosignatures, we need geosignatures. We need also understand if there's what is the surface composed of? How much volcanism is there? So how can you address this with a balloon mission?
Good question. The question is, how can we do a balloon mission not knowing much? Here we're going, well, the former Soviet Union sent 2 balloons to Venus. They lasted for 2 days each, just on their own battery power. So we're not so much worried about the coupling. We just don't want to land in a Hadley cell. But other than that, we're good. And one of the things I didn't mention is there's NASA and ESA missions that are going to answer all those bigger picture questions about the whole planet context. We're only going after the cloud droplets. We want to send a mass spectrometer and look for complex molecules. So our goal is to go to Venus, last about a week at a fixed altitude, and get the job done. Wait, but one more little thing. We're also trying to deploy these mini probes and imagine missions of the future that have lots of tiny little probes, like this big. And you can imagine those having an anemometer, so maybe they could give you useful information that you wouldn't otherwise get.
Slightly philosophical question about the false positives. So I think in your given time of life, you can only make sure that certain numbers of false positives are not the case. So then you can say, okay, fine, there is no false positive, but in the end, it's an infinite possibilities, right? So we don't really know. We are now doing this project. We actually are one of these nasty people who claim there might be another way to explain certain chemical observations. So, I mean, it's an infinite amount of false positives. How can we deal with that?
There's an infinite amount of false positives. How can we deal with it? I mean, we could list out like our top 10 false positives and through the modeling show how plausible they are. But the bottom line is we want to have hope. We want to have signs of life so the search can continue to more sophisticated telescopes like the ones I ended with.
Yeah, I think the plausibility of false positives is also an important thing to take into account. And I'm thinking back here to the announcement of the discovery of the very first exoplanet, 51 Peg. Mm-hmm. And the community spent a lot of time doing due diligence and trying to shoot it down, if it was possible that there was some previously undiscovered form of stellar pulsation that could have been giving rise to the periodic signal that was being interpreted as a planet. But in the long run, A, they couldn't find one, and B, everybody sort of knew that planets have to be there anyway, otherwise you couldn't form stars, because they have to dump their angular momentum somewhere. So in the end, I think it was plausibility arguments that won it over in the end, plus the fact that there were other teams that had been sitting on their data and who got round to actually analysing it and then had independent confirmation.
Right. I want to build on that because it'd be remiss of me not to mention methane. Methane is made by life on Earth. It also comes out of mid-ocean ridges and other places. But you know, the web is so good at finding methane. It is great because there's like giant features of methane. So there have been people wanting to promote methane, that even though it is made in other ways, that one could argue if you can see it, it has to be produced in such great amounts that these other infinite number of ways, or not infinite, I'm just picking on what you said, maybe less, maybe ruled out because we can't find a way to generate so much methane.